begin to look at the physiology of muscle contraction. And so we'll start to see what's occurring physiologically when we want that muscle to actually contract. And so during contraction itself, we had talked about the movement of all of our components within our sarcomere simply sliding past one another. Nothing changes size or shape as it does so, and this is our sliding filament mechanism or sliding filament theory. And so what we end up having is where those myosin heads attach up to the actin. And so they're going to grab on up there and they're going to start to pull those thin filaments in towards the M line. And so we're going to see a change in size of things like our I band and our H zone during muscle contraction. So the two of them are going to become smaller and smaller until during maximum contraction, those two are gone. The only thing that stays is the A band. And so that helps to show us that we don't have a change in the actual size or shape of the proteins themselves. What we see is a change in their movement, and that's it. So how do we get contraction? What's causing this muscle to contraction? We said that the muscle itself was excitable. It has the ability to carry a charge and to move an electrical impulse along it. And so how does that actually occur? It begins with a neuron. And so a neuron sends off a nerve impulse. And when that neuron's impulse gets to its own axon terminals, it then releases acetylcholine or ACH. This acetylcholine floats across that neuromuscular junction, moves down towards the muscle, and binds to receptors on the muscle's sarcolemma. Those receptors have sodium channels. And so there's sodium channels inside there. And so when we have this situation where we have a receptor on the surface of the cell, and as a acetylcholine molecule binds to it, that causes the receptor to then open up. And so as long as that acetylcholine is bound to it, it's going to open up that channel, and then sodium can rush into the inside of the cell. And so it's going to move to the inside of the cell. And that change in sodium, in terms of it's going from the outside of the cell to the inside of the cell, takes a relatively negative inside of the cell compared to the outside being positive. And since sodium is positive itself, that brings up the charge on the inside of the cell. And so it changes the relative potential charge there on the inside, and the inside goes to a positive. And so it's going to change the charge that is inside there. That gets propagated along the surface of the cell and spreads across the sarcolemma, goes down the T tubules to where we have our sarcoplasmic reticulum. At the sarcoplasmic reticulum, that change in charge opens up calcium channels, and those calcium channels now diffuse into, or allow calcium to diffuse into the sarcoplasm. And calcium is then going to go and bind to troponin. And so troponin and our tropomyosin complex, the calcium works like a key, the troponin is like a lock, and then the tropomyosin is like a door. And so it's going to allow for the three to interact with one another, and that's going to open up the binding site on the thin filament. And so that's going to allow for the thin filament to expose its binding site, and myosin now bind to it. And so just a little bit of a picture here to show the same process. And so we have our neurotransmitter coming down from the neuron, and so it's releasing its acetylcholine there. It binds to receptors. The inside of the cell is normally negative, and that causes the inside of the cell to turn positive, and that then travels along the surface of the cell, comes down the T tubule, and when the T tubule gets to where the sarcoplasmic reticulum is, and where we have these large expanses here, these are what are called the terminal cisterns, and those terminal cisterns then open up, calcium floods the inside of the cell, and that calcium is going to come over here and bind to troponin. When it does so, that changes the shape of troponin, and that moves tropomyosin out of the way, and you can see on these little purple actins here, there are little black dots. Those are the binding sites for our myosin head. And then we're going to start to go through the contraction process. So a little bit closer up of the actin, troponin, and tropomyosin. So the blue ones are the actin. We then have troponin, that's there, and where that calcium is going to bind to the troponin on there, and so it's going to bind to our troponin C. That's going to drag the three other components of troponin out of the way, and so that's going to move and change the shape of that troponin, and as it does so, it's going to drag our little tropomyosin out of the way, and so it's going to basically move it across there, exposing our binding site, and now myosin, which has been sitting there in a sense kind of laying in wait, as soon as that binding site is open, it can bind to it, and then it can go through the contraction process. At the neuromuscular junction here we have our receptor, and so we have a sodium channel that is with our acetylcholine receptor, and as soon as that acetylcholine binds to the receptor itself, that's going to allow for that channel to open up. Sodium rushes into the inside of the cell. The inside of the cell goes from being negative on the inside to being positive, and our muscle action potential begins, and then it's going to spread across the sarcolemma out of the motor end plate there and down the T tubule to the sarcoplasmic reticulum. So contraction itself is a series of 4 events. And so essentially it begins before we're ready for contraction to occur, because we have to do ATP hydrolysis. And so essentially this ATP hydrolysis is going to provide the energy to get the myosin ready to go. And so this is going to provide that energy for that. And so ATP hydrolysis has to happen before the actin is even ready for contraction. Once that binding site is open, the myosin binds to the actin, and we form cross bridges. And so that formation of the cross bridge is essentially the attachment of the myosin to the actin. It then goes through a power stroke. So this is the actual movement process. The myosin is going to change shape, and then we're going to get the detachment of myosin from the actin, and this isn't going to occur when a new ATP comes along to bind to the myosin. So it's going to come along and help to detach the myosin from the actin itself. This whole process will keep repeating and repeating and repeating as long as we have basically the elements needed for contraction. So we need ATP and we need calcium. And so ATP and calcium are two of the main components there, as long as we're still sending the signal to the muscle to get contraction to occur. And as long as we don't run out of ATP, where the muscle is going to start to become fatigued, we're going to have contraction keeping on happening within the muscle itself. So the contraction process begins with ATP here, and ATP comes along and it binds to our myosin. The ATP goes through hydrolysis and we get ADP and inorganic phosphate. That breaking of the phosphate bond gives the energy to take the myosin and bend it back down in a sense. And so it's going to recock the myosin head. If a binding site is available, the myosin head will form the cross bridge, and so it'll go back up and grab onto the actin. It then goes through its power stroke, and so it's going to go from essentially kind of being like this, being something like this, to being something like this, where the myosin head has pivoted and it's pulled and turned the actin towards the M line of the sarcomere. New ATP comes along, and so we're going to get more ATP coming in. It comes and binds to the myosin head. When it does so, that weakens the bond between the actin and the myosin, causing it to release, and then ATP can then be rehydrolyzed again, and we start the whole process over again and again and again. And this has happened during muscle contraction. So the myosin heads become activated, they become energized by ATP. So ATP has to be hydrolyzed, and we get ATP hydrolyzed into ADP, inorganic phosphate, and the energy that was liberated by that process. And that's what's going to, the energy is going to be imparted into the myosin head. It becomes a process where we transfer the energy itself into the myosin head that's there. The myosin head can then use it for the power stroke, and so it can pull that actin towards the M line. The whole thin filament begins to slide past the thick filament, and then new ATP comes along. It binds to the myosin head, that weakens the attachment to the actin, and we then get the whole process repeating again. We start back over at ATP being hydrolyzed. That hydrolysis gets us our inorganic phosphate and ADP once again, and energy can be imparted into the myosin. As long as our ATP is available, and as long as we have enough calcium in the sarcoplasm to keep those binding sites open, this process will continue to go on and on and on and on. If we start to run out of one of the substances that we need, this is where fatigue starts to set in for the muscles. So just kind of a quick overview from the beginning to the end. So we have our action potential coming down our neuron. From there, that causes the release of acetylcholine onto the receptors. Sodium rushes into the cell. That action potential travels across the sarcolemma, down the T tubules to our sarcoplasmic reticulum, where we have our terminal cisterns. Those terminal cisterns then release calcium that floods the inside of the cell. And so in terms of that quantity that's there, stored calcium inside the sarcoplasmic reticulum is about 10,000 times more concentrated than it is in the sarcoplasm normally. So there's a lot more inside there than what we normally have. So when we release calcium, it floods the inside of the cell with a just huge amount of calcium. Those go out and bind to troponin. Troponin moves tropomyosin out of the way. All of our binding sites are now exposed. Myosin can grab onto them. We get contraction. Our myosin goes through the power stroke. New ATP comes along. We hydrolyze it. We get our ADP and inorganic phosphate. And as long as that complex is essentially open, the muscle is going to continue to function. If the calcium has been pumped back into the sarcoplasmic reticulum, then that concentration goes down. The troponin changes shape back to its resting state. Tropomyosin recovers up all of the binding sites, and contraction cannot occur anymore. So that's essentially the relaxation portion. And so normally acetylcholine has been released out into that synaptic cleft, and the fate of the acetylcholine is it can float away out of the synaptic cleft. A large majority of it is broken down by acetylcholinesterase. So we have an enzyme to break down the acetylcholine that's there. Once that happens, the muscle action potential ceases. So the inside of the cell starts to go from being positive to being negative again. And so we change the charge inside of the cell once again. That closes the calcium channels on the sarcoplasmic reticulum. So if calcium is no longer being released out, now the calcium pumps, which are constantly running, are going to pump calcium back in, and the calcium is going to get into sarcoplasmic reticulum, and it's going to bind up to a protein called calsequestrin, and that's what's going to allow us to have that 10,000 to 1 concentration between the cytoplasm and that of the sarcoplasmic reticulum. With the calcium gone, our tropomyosin troponin complex recovers over the binding sites all over the actin, and now contraction can no longer occur. At the neuromuscular junction, we have our neuron, and that neuron is going through basically the same kind of a process. The inside of the cell is going from a negative to a positive, and so it's going through an action potential as well. And when we get to the end of the neuron at these neuromuscular junctions, the synapses that are there, calcium starts to flood the inside of our neuron, and that drives our vesicles towards the end of the neuromuscular junction there. So it comes to the axon terminals there, releases acetylcholine out into the synaptic cleft, and that then binds to our receptors. Those receptors then open up, sodium rushes to the inside of the cell, and we have a large influx of sodium there. A huge amount of receptors, which means that we also get a huge amount of acetylcholine being released into the synapse itself as well. And that's going to lead to very quick contraction of muscle. Once that action potential has come to the neuromuscular junction itself, and we get the release of acetylcholine, the acetylcholine goes out and binds to the receptors. On the motor end plate there, where the actual receptors themselves are, sodium rushes into the inside of the cell, takes the inside of the cell from being basically negative and turns it into a positive. And so it's going to go from a relative negative voltage to a relative positive voltage, similar to that of a battery. We have a negative side and we have a positive side. And so that occurs on the inside and outside of that cell. And then that impulse is going to travel along the whole surface of the cell until it gets to the sarcoplasmic reticulum. And at the sarcoplasmic reticulum, calcium then releases our calcium. Calcium binds to the troponin, moves tropomyosin out of the way. Almost as soon as acetylcholine is dumped into the synapse, acetylcholinesterase, ACHE, is going to come in and start to break it down, unless another impulse comes along and causes the release of more acetylcholine. We're going to get relaxation of the muscle. Since we have this series of events occurring at the neuromuscular junction, we have the potential to alter it. And so we can change what things are going on there by either stopping the release of our acetylcholine from the get-go. And so we just block its release. And this is what botulinum toxin does. And so botulinum toxin comes in and blocks the actual release of our ACH, and that blocks the release of the acetylcholine there. And when that does that, even though the person is telling their neuron to fire and release acetylcholine so that we can have muscle contraction, if we don't get any acetylcholine released, contraction cannot occur. And so that doesn't occur in there. Botulinum toxin is something that is found in canned food at times. And so a can of food that is kind of bulging out is a good indicator that there's botulinum toxin being produced inside there from the bacteria themselves. And so that's one of the ways that we can acquire it. Botulinum toxin is also the same toxin that is used for Botox. And so not only do we can we get it from a kind of natural source, we can also inject it into ourselves, and it causes the same paralysis in the muscles that are there. If we have eaten the botulinum toxin, the thing is going to cause paralysis all over the body, and the most important muscle to not cause paralysis in is the diaphragm. And so that's where we're going to have this problem in breathing if that botulinum toxin spreads to the diaphragm itself. Curare is another substance, again, a natural substance. This is the same stuff that is used from essentially poison dart frogs. And so the paralysis occurs here by having the acetylcholine receptors blocked. And so now we get the acetylcholine released. There's still acetylcholine coming out, but it can't go anywhere. It's got nowhere to bind. So as far as the muscle is concerned, nothing happened. The neuron is still doing its job the way it's supposed to, but now it has nowhere to bind up that acetylcholine. This was one of the first substances used to cause paralysis for surgery. One thing that can be used to reverse the process of having those acetylcholine receptors blocked is to have an anticholinesterase agent. And so neostigmine is one of those. And basically what it does is it blocks the activity of acetylcholinesterase. And so if we're not breaking down the acetylcholine, we're getting more and more acetylcholine being available in the synapse, which means that that's going to essentially strengthen the signal that's being sent. And it can be used as a process to increase muscle contraction in diseases like myasthenia gravis. That's there. And then it'll also reverse the effects of a paralytic that is causing blocking at the receptor level. When we have the release of acetylcholine, and then we get the release of calcium out into the cell, calcium itself needs to be removed from the sarcoplasm in order to get relaxation. And so when we die, rigor mortis sets in somewhere in the neighborhood of a few hours after death to, if it's the body is really cold, it may take more than that for it to occur. Ambient temperature plays a little bit of a role in when that rigor mortis sets in. And essentially what's happening is when we die, the cells themselves don't necessarily know that things have stopped. And so they keep on doing their thing. So we still have calcium pumps running inside the muscle cells. We still have ATP available, etc. But after death, eventually those calcium channels stop functioning. And calcium starts to leak out into the sarcoplasm from the sarcoplasmic reticulum. Once calcium leaks out into the sarcoplasmic reticulum, it just goes and does its job. It binds to the troponin, moves tropomyosin out of the way, and now all of a sudden the myosin heads can bind to the actin again. And so that's going to cause one last muscle contraction in a sense. But since we've run out of ATP, we can't undo the process. And so ATP is not available to unbind our myosin from the actin, so that muscle contraction just stays. And the only thing that's going to stop it is the eventual breakdown of proteins inside the muscle. And so we have naturally proteolytic enzymes inside muscles, and that same process starts to occur when we want to break down muscle. So when we're not using a muscle so much, we're going to break down those proteins and recycle them. But in the dead cell, those proteolytic enzymes literally start to digest the proteins inside the cell. And so as those start to break, then the muscles start to relax. When we contract muscles, the muscles themselves have kind of some optimal portions as far as the length of the muscle when it comes to contraction of the muscle itself. And so depending upon how much overlap we have of the actin and the myosin dictates essentially how well the myosin can grab on here and pull the actin in. And so the more overlap we have, the stronger that contraction can become. So as we lengthen a muscle, as we stretch that muscle out, that means that there's going to be less overlap of those proteins, which means that we're not going to get as strong a contraction occurring. And so optimally, the muscle is going to be somewhere in the middle of its contraction. And so in terms of the middle of its length, in order to get the greatest amount of contraction available. And so the greatest amount of force being generated is in this area here. And so we have the greatest amount of force that can be generated. So if we were talking about something like the biceps brachii or so, we would want the muscle somewhere, or the elbow somewhere around 90 degrees in order to get the greatest amount of force we can. If the elbow is straightened all the way, we can't generate as much force. And so as the length becomes greater, we can't generate as much force. And as well, when we start to get that muscle shortened all the way, so when our elbow is at its maximum shortening, we start to lose contraction as well. We can't generate as much force. And so the greatest ability of that muscle to contract is somewhere within the middle range of the muscle. In order to keep the muscle going, we need its metabolism to essentially be maintained. And in order for that to occur, we need the greatest amount of ATP as we can get. And so we have three different processes that can generate ATP for the cells. We have creatine phosphate, we have anaerobic cellular respiration, and we have aerobic cellular respiration. So either one of those three can provide for ATP within the cell because we don't have a whole lot of reserve in a sense of ATP inside the cells themselves. So when we do have a large amount of ATP, and so we have an excess amount within the cell itself, that excess ATP is relatively unstable. So we're going to take one of the phosphates and we're going to donate it over to creatine. That's going to form creatine phosphate. And then that creatine phosphate, when the muscle needs to go to work again, it can release its phosphate group over to ADP again, and we get ATP. And now that's then going to get hydrolyzed. We get inorganic phosphate. We get ADP again, and that ADP can then be used to either take it off of creatine phosphate again, or it can be used and recycled elsewhere in the cell. If too much ADP starts to develop, this can start to lead to fatigue of the muscle as well. That's there. This creatine phosphate can only power the muscle for a relatively short amount of time. So we don't really get a whole lot of energy from creatine phosphate because there just isn't a whole lot there in the cells. Anaerobic respiration uses no oxygen. So in the process of forming ATP, we take our glucose and we're going to get 2 ATP from it. And so we're going to go through the process of glycolysis. That glycolysis is going to get us 2 ATP, and it's going to provide for now energy for the cell. We've basically doubled what we got from creatine phosphate, but this still isn't enough to run the muscle for very long. It also leads to the buildup of lactic acid, that burning sensation in the muscle when it's starting to tell you that I can't work anymore, because it's doing it in anaerobic respiration. So anaerobic respiration doesn't give us a whole lot of energy for the cell either. So we need something that's going to be able to power the cell for basically all day long. And that's where aerobic cellular respiration comes into play. And so aerobic respiration is going to give us, from a single glucose molecule, we're going to get 36 ATP. But even though we can use glucose, we can also use amino acids, so we can break down proteins. We can use fatty acids, we can break down lipids, and so we can break down adipose, that's there. Or we can use glucose, and so we can get that glucose coming into the cell itself. And so we're going to go beyond just simple glycolysis and go through the rest of the aerobic cellular respiration, and that's going to get us the greatest amount of ATP. The majority of the time, our muscles are working with aerobic cellular respiration. So they're going through the aerobic process itself. But when we start to use the muscle over and over and over and over again, this is where we're then going to start to move into anaerobic processes, and the muscle is going to have to slow down or stop in order to catch up. So in summary, creatine phosphate gets us a single ATP. Aerobic cellular respiration is going to get us the greatest amount at 36 ATP. Anaerobic respiration is going to get us 2 ATP, but the benefit to both creatine phosphate and anaerobic is that both of them require no oxygen. But we do need oxygen in order to get us the greatest amount of ATP within the cells themselves. And that's going to allow us to power the muscle essentially for all day long. If we run out of any of the components that we need for muscle contraction, we're going to get muscle fatigue itself. And so this is where the muscle is going to start to fatigue because we've run out of calcium, because we've run out of ATP, because lactic acid has been building up because we're running out of ATP, because ADP is building up. All of those are going to add to the fatigue of the muscle itself. If we can't get oxygen to the cells, so there's a cardiovascular issue, there's a respiratory issue, that can lead to fatigue of the muscles as well. We can also get a process where we have a lack of energy. No more glucose, no more glycogen in the cells themselves. Once that muscle has been worked and worked and worked, and we've used it and we've fatigued that muscle, we're going to go into an oxygen debt. And so this is where essentially all the normal processes that we have to use for the muscle need to kind of be paid back. And so the oxygen reserves, and so we have essentially similar to what we have in red blood cells. In red blood cells, we have hemoglobin. In muscles, we have myoglobin. That's going to bind up some oxygen inside the muscle. We've got to get the lactic acid out of the cells, and so that either needs to be converted into pyruvic acid or leaked out of the cells and into the bloodstream. We've got to pay back the glucose that was used out of the muscle. So we need to re-get glucose in excess so that we have glycogen. But in order to do everything that we need glucose just for ATP. And so glucose is going to have to come in first to replenish all of our ATP back into the cell. Once all of that is replenished, then we've paid back our oxygen debt. When a muscle starts to work during exercise itself, essentially oxygen to that muscle comes from either myoglobin, where we can store oxygen molecules inside the cell, or primarily it comes from the blood. And so blood flow to the muscles is going to allow for us to give the cell all the ATP it needs by going through aerobic cellular respiration. And that's going to allow the muscle to work there. As we need that muscle to continue to work, we know that as we start to exercise, our breathing rate goes up, our heart rate goes up, and all that is in response to getting the oxygen to the muscle. And so we're going to have that change there so that we increase the amount of blood flow with adequate amounts of oxygen to the muscles themselves. Once we've done that exercise, those oxygen levels are going to stay elevated. Just because we stop running doesn't mean that our breathing rate goes down, doesn't mean that our heart rate goes down instantly. We've got to pay back the oxygen debt that's there. And so that's going to allow for that to occur. One of the potential benefits of increased temperature when muscle contracts, that byproduct of breaking ATP leads to heat. And what that allows for is all of our chemical reactions to move faster. And so we have the benefit of increased temperature when we have muscle contraction as well.